Hello friends, welcome back to my YouTube channel, The Zone of Anatomy. And today we are going to discuss about function of synapses and neurotransmitters. In this we see what are the nerve fibers, its type, what is neuromuscular junction, how the impulse get transmitted and many more. So let's begin for today's lecture. Coming to the type of nerve fibers, let us first define what is nerve fiber. Nerve fibers are basically the names which is given to the axon or the dendrite of the nerve cell. We have already known what is the nerve cell, what part the nerve cell has. So the nerve fibers is the part or the name given to the axon or a dendrite. When we are talking about the type of the nerve fibers, we are talking about 5 type of nerve fibers that are structurally we see what are myelinated and non-myelinated. Then we see functionally, that is motor or sensory. Then we see developmentally, that is somatic or autonomic. Then chemically, that is androgenic or cholinergic receptors type. And physiologically, we see there are 3 type of fibers: type A, type B, type C, and their further distribution. So amongst these all, physiologically, nerve fibers are most important. Coming to the nerve fibers, as I've already say, structurally we have myelinated and non-myelinated. The myelinated fibers are those which has the myelin sheath, and the non-myelinated is the one which do not have the myelin sheath. Then functionally we have the motor we say the efferent, and another we have a sensory or the afferent. Then developmentally we see that it is somatic or the autonomic. Chemically, as I say, it is adrenergic and cholinergic. What is adrenergic? It is a neurotransmitter in noradrenaline, and cholinergic is the neurotransmitter in acetylcholine. We also going to see how the acetylcholine work in the further slide. Then we come to physiologically. Physiologically, we have actually 2 types. One is sensory, one is motor. And at the we also see that nerve fibers which have only like sensory nerves which have only and only like classified on numerically based. So we'll see further. Physiological classification, as I said, is one of the most important. We are talking here about those fibers which has both sensory as well as the motor fibers. We have type A, that is myelinated. MCQ come from here. Type A is 3 type: alpha, beta, gamma. Alpha is for the proprioception and somatic motor function. It's not somatic motor. We can say also it is, okay, we can make it as somatic motor function. That will be more better to understand. Then we see beta, that is touch and pressure reception. Then we have a delta, that is pain, touch, cold, and temperature. So the question, like this slide is very important. The type A is the type of fibers which has myelin sheath over it. It has 3 type: alpha, beta, gamma. When we talk about type 2, it is preganglionic autonomic, that is, it is also myelinated. So the MCQ come on this myelination only. Type C is non-myelinated, that is, it do not have the myelin sheath. MCQ, it is present in the postganglionic synaptic and the dorsal root. In dorsal root is concerned with basically our pain, temperature, mechanoreception, reflex responses. So the question may ask that the mechanoreception is like mechanoreception is concerned with fiber, that is type C. Then we have a touch sensation is by which fiber, that is type A. So just learn these things. When we come to sensory nerve fibers, I have already said that we mentioned classify them numerically, that is 1A, 1B, then 2, then 3, and then 4. Moving forward, conduction of the impulse through nerve fibers. Now we are going to see like how these impulse get conducted through the entire nerve fibers, after which we see its property also, how or what properties it has. So let's quickly discuss what are the properties of the nerve fibers. Basically, the nerve fibers is concerned with our excitability, conductivity, then it has some adaptation, it has submission property. So basically you need to learn the property of the nerve fiber, that is excitability, conductivity. That's the only thing you need to remember in this. Then when we talk about the conduction, like how conduction is taken place. So we have normally divided the conduction in two phases. That is, one is continuous conduction, which you see in this non-myelinated axons, and the solitary conduction, which you see in this myelinated axon. So one MCQ come that the continuous conduction is in non-myelinated axon, and the solitary conduction is in myelinated. So one MCQ is from here itself. Coming to what is this continuous nerve impulse? Continuous nerve impulse is basically when we talk about the action potential or the nerve impulse, it passes continuously without any break along this axon. And then suppose this one is excited, then it excites another, then another, then another, then another. Here you can see the arrow also. So continuous conduction means that the action potential passes continuously without any break along the axon, and it excites the neighbor areas of the membrane also. Once it is initiated, once the stimulus come, then it propagates continuously without any stoppage. So action potential are propagated continuously, and the conduction is continuous, we also termed it as the local currents. So in non-myelinated, continuous conduction of nerve impulse is seen, and in myelinated we have the solitary conduction of impulse. In this, the myelinated nerve fibers can be stimulated only by these gap, that is the node of Ranvier, where the axon, you can see here that the axons is naked at the point of node of Ranvier, and the ions here can pass freely through the plasma membrane between these ECF and the exoplasm. So from the node of Ranvier only the impulse is transmitted. So we can say that the action potential jumps from one node to the next node and so on. So the action potential at one end sets up the current in the surrounding tissue fluid, and quickly then it depolarizes the neck, then depolarizes the neck, and so in the myelinated axon, we can say that there is the jumping of action potential or the nerve impulse from one node of Ranvier to the next node of Ranvier, and this jumping of impulse, we termed it as a solitary, and the conduction is termed as solitary conduction. So we can say that there is a jumping of stimulus. So this myelinated axons has more fast conduction as compared to the non-myelinated conduction. I hope this is clear to every one of you. Then when we talk about the factors which determine the speed, we say that in the myelinated it is a fast conduction, and in non-myelinated it is a slow conduction. So how we define this? We define this by these parameters, like the thickness of the fiber, how it is affect the thickness. The thicker nerve fiber conducts rapidly as compared to the thinner one. When we talk about the presence of myelin sheath, we see that there is the solitary conduction. So due to the presence of myelin sheath, the action potential jumps, and the axons can conduct 40 to 50 times faster. So the faster conduction is due to the presence of myelin sheath. Then the temperature also, when the temperature is cool, so it decreases the conduction, whereas the temperature is high or at the hot temperature, the conductivity of the nerve impulse increases. When we talk about the mechanical resistance, so the mechanical resistance decreases conductivity as well as excitability when we come about the blood supply. So if, for example, we have some artery or vein and the supply, blood supply, we cut it. So obviously it diminished the the properties of nerve impulse is lost. So that's sometimes blood supply also plays a major role. Then chemicals like we have narcotics. So narcotics, we normally say that taking these narcotics diminish the conduction of nerve impulse. So when coming to the hydrogen concentration, the more the H plus concentration, the less is the conductivity. And when we come to the effects of ions, in further slide also we see these points. So just this is more important from MCQ point of view. If we have a lack of calcium, then there is increase in excitability. And if we have a increased of calcium, we have a decreased excitability. So calcium and the excitability are inversely proportional to each other. Coming to the conduction, we have already seen, I have already explained in the previous slide what is continuous conduction, what is solitary conduction. As I say, continuous conduction is in unmyelinated nerve fibers, MCQ. Nerve passes along axon. It is a slow mechanism and it is like local current. The nerve impulse are conducted by local current. Coming to the solitary conduction, myelinated nerve fibers, MCQ again, jumping of nerve impulse, another MCQ, and it is a rapid mechanism as compared to the continuous conduction. Now moving forward, there is a few things which we need to, just remember what are these terms and nothing more to study about in this lead. First is what is Hush factor? Hush factor is basically the velocity, or we say that the diameter ratio of the peripheral nerve fibers. Okay, so how we take this factor is basically we see the velocity of the nerve fibers conduction and we know the diameter of the nerve fiber. So we put the velocity and the diameter directly proportional to each other, and whatever the constant come, it is known as the Hirsch factor, and the Hirsch factor for the mammalian is 6. So these 3 points you need to remember. Then coming to what is the strength duration curve. What is strength duration curve? It is basically the relationship between the strength of the current and the duration of the stimulus is called the strength duration curve. So in this we need to just think or know one thing: the more the strength, here you can see the less is the time, and the less is the duration, the more is the less is the strength, or it is basically the less is the strength, the longer the duration. We have less strength over it, but we have a longer duration. Okay, and more strength, we have a few duration. So more the strength, less is the duration, and we do the vice versa. Coming to the myelination, what is now the myelination? Myelination is the process of formation of myelin sheath. I think 1 second. Okay, yeah, here we have one more thing, that is the neurotrophin. It is basically the protein necessary for the survival of neurons, that is functional unit of nervous system, and the neuroglia cells, that is are supporting cells of the neurons. And these three are nerve growth factor, brain derived neurotrophic factors, and neurotrophic. These are just to remember, not to go deeply in these, as it is not much more important. Then we need see what is the myelination. As I say, myelination is the process of formation of myelin sheath around these nerve fibers. And myelin sheath, whenever someone asks when the myelin sheath started to begin, it begins at the time of the birth. Coming to the mechanism, we have see two parts in this. One is peripheral nervous system and we see the central nervous system. From peripheral nervous system, the question arises, the myelin sheath is formed by Schwann cells. In central nervous system, the myelin sheath is formed by oligodendrocyte cells. You can see these oligodendrocyte cells form these sheath. On the peripheral nervous system, we see only these Schwann cells to form a single nerve fiber, whereas in the central nervous system, we see many and many nerve fibers coming out from the oligodendrocyte. So, when we see how these myelination formation of myelin occur, first we see in the peripheral nervous system. In the peripheral nervous system, what happened? That the axon is naked. So firstly, we have our axon that is totally naked. It do not have any myelin. Then it is accompanied by a linear row of Schwann cells. You see these Schwann cells coming over here, here, here. So Schwann cells come over this. Then each cells completely, it is like totally completely invest the axon, or we can say that the outer membrane and the inner membrane of the Schwann cells usually become the double folded. These become double folded, upper part and the lower part, and we call them as the mesoaxon. And then the Schwann cells complete its wrapping all over it, and we say it as a myelin sheath in the peripheral nervous system. So we just need to remember 3 points. First is Schwann cells. First, our nerve fibers is naked. Schwann cells is important in this. Third, the formation of mesoaxon, and fourth, the myelin sheath formed by wrapping around this. Coming to the central nervous system, in the central nervous system, the oligodendrocytes takes part in it. The process, this pairing process is similar in central nervous system also. But the difference in peripheral nervous system and the central nervous system is only one, that in the central nervous system, the process of this single dendrocyte can deposit myelin around several axons. But in peripheral nervous system, each fiber has its own Schwann cells, and in central nervous system, only one oligodendrocyte makes all these nerve fibers. So that is for the myelination part. So here I have put all the points, whatever I have explained it to you. This is a process of formation of myelin sheath and around the nerve fibers. It comes in the MCQ for the definition of the myelination. It increases the velocity. Why we do the myelination? Because it increases the velocity, as we have already seen when we talk about the conduction. Then peripheral myelin sheath is concerned with Schwann cells. CNS myelin sheath is concerned with oligodendrocytes. Then its mechanism I have already explained in the previous slide, so you can go through these steps once again. Now we come to the neurotransmitter. When we talk about the neurotransmitters are basically the chemical agents that are released from the nerve ending part and then transmitted these nerve impulse through the synapses from nerve to nerve, or we say that the nerve to nerve communication is done by the chemical agent, that is neurotransmitters. So it is the chemicals that are present in the synaptic part. Coming to neurotransmitters, we can first see what are the type of the neurotransmitters. Neurotransmitters can be 3: excitatory, inhibitory, or we can say both excitatory and inhibitory. These two tables are very important because it comes in your it helps in your GIT tract also. It helps in your respiration part, in cardio part also. So excitatory, it is epinephrine and glutamate. Inhibitory is GABA. Then dopamine, serotonin, mood changing hormone, which we say glycine, aniline. Then acetylcholine, norepinephrine are the both, that is excitatory and inhibitory. Then histamine, prostaglandins, and 5-hydroxytryptamine. That is 5-HT we normally mention, that is 5-hydroxytryptamine. That is the only thing we need to learn in this slide. What are excitatory, what is inhibitory? Like dopamine, GABA, serotonin, we all know it is inhibitory. Epinephrine is excitatory always, and both can be acetylcholine. Coming to the neurotransmitter that conduct rapid and the slow transmission. So first is our acetylcholine. Actually, when we talk about acetylcholine, it has an excitatory effect, but it also has sometimes the inhibitory effect. So we put it in category of this both. So it conducts the rapid conduction. Say, then we have the amines in this part. What are the amine neurotransmitter? That is epinephrine, norepinephrine, dopamine, serotonin, and histamine that conducts rapid neurotransmitter. Amino acids derivatives can be GABA, glycine, glutamate, aspartate, and nitric oxide. Slow conduction is done by normally the glands, that is hypothalamic hormone secreted by, that is somatotrophin and thyroid releasing hormones, and pituitary peptide, ACTH, TSH, LH, GH. So these things all we know, like in the peptides of gut, I already say that neurotransmitters help in our GI tract and brain also. So we have already familiar with these words, that is gastrin, substance P, that is VIP, that is insulin, glucagon, CCK, Just memorize these table. It is the only important table you can learn and direct question can come. When we talk about now, when we talk about what are the criteria of the neurotransmitter, so the criteria is very simple, that the chemicals must be present in the presynaptic elements. Okay. Then they are synthesized from precursor substances. They have this synthetic enzyme that is located on the presynaptic. This is presynaptic, this is postsynaptic. So the enzymes located in the presynaptic side of the synapse, then they remain stored in the synaptic vessels. See, these are the synaptic vessels where neurotransmitters can be stored. Then they have a postsynaptic receptors, and the chemicals, like whatever the neurotransmitter, like the ACH come, it binds over it, and then the inactivation is present in the biochemical inactivation mechanism occur. So these are the criteria on which our neurotransmitters work. Now we see, if we remove the neurotransmitter, what have, or we can say how is neurotransmitter removed from the synaptic cleft. We need to remove these neurotransmitters from the synaptic cleft. What it can be do? First, what happened? As we know that there is the stimulation of the postsynaptic receptor by the neurotransmitter. Neurotransmitters first, stimulate this postsynaptic neurons. You see, this is neurotransmitter then come here and then stimulate this postsynaptic receptor. These neurotransmitters then need to be, removed rapidly from this neurotransmitter cleft, because in order to repolarization to occur. So when we see why we need the removal of neurotransmitter, it is basically due to we need the repolarization to occur. So that's why we want this removal of neurotransmitters. So now when we see what is the complete process of this, first we say that there is the enzyme destruction of the neurotransmitter. For example, if we have suppose an acetylcholine and it is removed from the synaptic cleft, see, this is our acetylcholine, the purple one, and it is releasing from this synaptic cleft. So by hydrolysis of acetylcholine, so acetylcholine get hydrolyzed into two parts, that is choline and acetate, and the reaction catalyzed by the enzyme, that is acetylcholinesterase. So we need to remember two things. What? That the acetylcholine has divided into two parts, or we say that the acetylcholine is made up of from two parts, that is acetyl-CoA and an acetate, and the enzyme responsible for the destruction of this is acetylcholinesterase enzyme. Second part is we need to reuptake. What this reuptake means here, it is basically a process by which we normally transport that, or we say that the neurotransmitters are transported from these synaptic clefts back into the cytoplasm of the neurons. So reuptake means transport of the neurotransmitter from the synaptic cleft into the cytoplasm. Then we have the diffusion into the surrounding fluid. We say that our neurotransmitter has already gone in the cytoplasm. Then these neurotransmitter which is present in the cytoplasm binds with our postsynaptic and presynaptic receptors. So that's the complete removal process. Coming to how acetylcholine mechanism, as we have seen when we already discussed what is biosynthesis of it, that is acetylcholine is synthesized in the plasma of our axon, and it is made from two parts. What two parts? Acetyl-CoA plus choline, and it forms acetylcholine, and acetylcholine then break down into two parts, that is choline and acetate. So now we have this biosynthesis with that acetylcholine esterase convert acetylcholine into choline and acetate. When we talk about its mechanism is very easy. That is, first we have the arrival of impulse in the nerve end. We know acetylcholine is present at the nerve ending of the fibers. Then there is the opening of the calcium channels. Then when the calcium ions enters into the nerve ending, the vessels containing these ACH fuses with the presynaptic vessels and release of the acetylcholine by the exocytosis. I can once again repeat it by this part. So the mechanism is very simple, that if we have some impulse arises over here, we have impulse over it, okay, then there is the opening of the calcium. As soon as impulse comes, the calcium gated ion channels open and calcium starts moving in the nerve ending vessels. These nerve ending vessels contain the acetyl-CoA, and then these acetyl-CoA fuses with these presynaptic membrane and it releases the like acetylcholine in this synaptic cleft, and then there is the release of ACH, and the process of this release of ACH is known as exocytosis. So this is a complete process of how the acetylcholine work. Now we need to talk about, here is the process, whatever I have explained in the previous slide, here is the process. I make it in the flowchart form. Coming to the synapses, the part of the synapses, when we talk about what is the synapse, synapses are the site of the functional contact between what? Between the neurons and other effector cells, that can be muscles or our gland cells. It is known as the synapses. Then when we say what are the components or the elements of the synapses, it is presynaptic, synaptic, and the postsynaptic. Presynaptic is also known as the presynaptic nerve or the end feet, and it is basically the it has contain exoplasm, and the exoplasm has three components in it, that is neurotransmitter vessels, mitochondria, and lysosomes. When we come to the synaptic cleft, there is a synaptic cleft. We already seen the diagram, there is presynaptic part, then we have a space known as the synaptic cleft, and then we have the postsynaptic part. So the synaptic cleft has contained neurotransmitting degrading enzyme. Normally these neurotransmitting degrading enzyme is acetylcholine esterase that degrades our acetylcholine into choline part, and another part is acetate part. So this is we already discussed, and we have polysaccharides. Coming to the postsynaptic part, it has the receptor protein and the impulse generating part. It has the receptor proteins. Its function is to impulse generate. Coming to the properties of functions and properties of synapses. So what is the function of synapse? The basic function of synapse is basically transmission of the nerve impulse, whatever action potential comes from one neuron to another neuron, and ultimately to the effector cells, or we say the muscles or the gland cells. Then there is a formation of reflex arc, or another function can be integration of the nerve impulse. So these three are the functions of it. Coming to the properties of the synapses, properties is very way. First property is there must be one-way conduction. What is one-way conduction? That the conduction is always in one direction and not bidirection. That is from presynaptic neuron part to synaptic left to postsynaptic. Since axon can conduct that both the direction, like equal facility, the one way gate of the synapse is necessary. So we say that the synapse has a property of one-way conduction, that we first have presynaptic part and then postsynaptic part. So now we come to see what is synaptic delay. It is synaptic delay is the basically time lag or the time interval, or it is the interval of the time between this arrival of impulse over here at the presynaptic part and the outset or the result at the postsynaptic neurons. Then we come how fatigue is the property of this synapse. When a synapse is rapid or fast and stimulated continuously, like it is stimulating very fast and continuously, then the number of discharges from this post synaptic neuron normally decreases, and we call this fatigue. We see this in the exhaustion time. We see during then our muscles is continuously working. At that time we see that there is a fatigue in the muscle. That's the reason that the synapse is so continuous and rapid that there is discharges at the postsynaptic neurons decreases. Coming to another, that is synaptic response. At the synaptic, when we talk about the synaptic response, the impulse what is the synaptic response means? Whatever the impulse we receive, first we integrate that information and then we discharge, and that discharge lead to the action on our muscles and the glands. Then we have the inhibition property. Inhibition mechanism is associated with our presynaptic and postsynaptic part. Then inhibition can be postsynaptic and presynaptic. How initiation takes place in postsynaptic part? That is, if we have the calcium influx in the postsynaptic, suppose here in the postsynaptic part, okay, we have the coming of our calcium from this ion channel receptors. So automatically there is the efflux from the calcium. If calcium comes in, the potassium goes out, and by this there is the activation of receptor enzymes. Coming to another part, that is inhibition of presynaptic. If there is the inhibition of this, then calcium influx decreases automatically. If calcium influx in the presynaptic terminal decreases, then there is also decrease in the excitatory transmitter. This is one part. Coming to another, if calcium is influx, then potassium is in the action of efflux, and that decreases the calcium influx. So there is a direct inhibition of transmission release into excitatory ending. So this is the only thing which you need to remember about this. Now we are going to see how these, impulse is transmitted. First, whenever we see any action potential or the nerve impulse arrive here, in this are pre-synaptic part. So already we know this is pre, this is synaptic left, and this is post. So our impulse is coming here. Then as soon as impulse come, the channels over here opens. What channels? The calcium channels open. Then once the calcium channels open, then there is the entry of these calcium ions into the presynaptic vessels. You can see the calcium is coming in from the synaptic cleft. Then these calcium causes fusion of these neurotransmitter with this presynaptic membrane. So neurotransmitters are packed in this part. This is known as our synaptic vessels. So these neurotransmitters, these synaptic vessels get fused with this part. Fuse this presynaptic membrane. After fusion of these both, the neurotransmitters get diffused into the synaptic cleft. Here are synaptic cleft. Here our neurotransmitters are released in the space. This is known as synaptic cleft, and then these neurotransmitters binds with the post. You see, it is going to started binding with post part. Now we need to see if it binds with the post part, what actions we can see. First, the action which can be triggered, that is if there is influx of sodium. And there is two possibility. One, there is the influx of sodium, and one, there is the influx of chlorine ions. So when the sodium is coming in, then there is depolarization takes place, and when there is the chlorine coming in, there is hyperpolarization taking place. So you need to remember one thing, that the sodium is concerned with the production of the impulse, and the influx of chlorine do not produce any impulse because there is no action potential is generated due to hyperpolarization. So here is the story what we are talking about after the neurotransmitters binds with the post or synaptic receptors. Like neurotransmitters bind with the postsynaptic receptors, we see that these bind here with the postsynaptic receptors. Then there is two chances we have, that is influx of sodium and influx of chlorine ions. If we talk about influx of sodium, then there is depolarization, then the action potential is generated over there, and the nerve impulse production is conducted. And when we talk about influx of chlorine, then there is hyperpolarization part, and there is no action potential is generated and no impulse produces in this part. So no impulse production is concerned with chlorine, and impulse production in postsynaptic part concerned with sodium. Now, since we have done with our chemical synapses part, now we'll see what is neuromuscular junction. Neuromuscular junction is a specialized area where motor nerves are all the motor neurons get terminated, and they are devoid of myelinated sheath. Or we can say that neuromuscular junction is the area where a motor nerves get terminated on a skeletal muscle fibers, and the action potential get transmitted nerves to the muscle, the neuromuscular junction. The parts of the neuromuscular junction which we need to remember, first is these axon terminals, second is the synaptic cleft, and last is this motor end plate, which we also known as the postsynaptic element. As we say, neuromuscular junction is concerned with our skeletal muscle fibers and the action potential of it. So these are our motor neurons, these are our skeletal muscles, and these are our cleft, synaptic cleft, and similar story is going on presynaptic, postsynaptic part. Now we'll see how the, impulse or mechanism of neurotransmission is. So first we say here again the story begins the same. Arrival of impulse or the action potential at the axon terminal. Here we have the axon terminal we see in the previous slide. Then there is the opening of the calcium channels, which we have already discussed. Calcium moves into the axon part. Calcium causes fusion of, then calcium is moving in, and here is acetylcholine present in our synaptic vessels. It get released, it get fused with this membrane, and then there is the release of acetylcholine into the cleft, synaptic cleft. Once this acetylcholine is released, then acetylcholine, whatever it is released, it gets bind with the acetylcholine receptors, which is present on our motor end plate. This is our motor end plate. The motor end plate this is, and here we have a acetylcholine receptors present. Then there is the opening of the acetylcholine channels. Why? Because there is a receptor for acetylcholine, and acetylcholine needs to go over here. Then there is similar story going on. Similar. If there is influx of sodium, then there is the generation, or we say end plate potential is generated, and then it causes the movement of our skeletal muscles. So that's the complete story. You need to just know the mechanism of synapses, and you can crack any part of neuromuscular junction. So this is the point which I have mentioned, that first there is the arrival of axons, then opening of calcium, then our calcium and this what we say vessels, synaptic vessels fuses with the membrane, acetylcholine releases, acetylcholine release and binds to the receptors present on the postsynaptic cleft, and then here their sodium channels get open. Once the sodium ion gets open, acetylcholine through it, and then the action potential is generated. So guys, that is from my side on this topic. It is very basic and many questions may arise on the neurotransmitter part. So just go through it very deeply and just study whatever I have explained in the lecture. It might get help you. If you like the content of my video, do like, share, subscribe, and do put your valuable reviews in the comment section, so I can come to know you are liking my video, or whatever changes you need, just put in the comment section. And the extra sheets or the extra points which we need to remember, I have put it in the Telegram group, and the link for the same app I put it in the description box. So just go over there and see all the extra study material, or you can practice over there also. And if you haven't seen our previous lectures on the neurophysiology part 1, which comprises of our brain and other things, you can go through that once again. I have put the point or the link in the description box. So before starting this video, make sure you go and see what is the central nervous system and what is the parts of the brain. So that's from my side. We'll meet in the another lecture. Thank you.